The structure of DNA is actually a fairly simple thing to understand. But to begin with this, we need to start off with what are the building blocks of DNA? The building blocks of DNA are called nucleotides. Let's take a quick look over here, and you can see the basic structure of a nucleotide. All nucleotides have three basic components. There's a phosphate group, which has a negative charge. There's a pentose sugar, which simply means it's got 5 carbons. They're actually numbered. This is carbon 1, 2, 3, 4, and carbon number 5. So the phosphate's attached over here to carbon number 5. And then over there, you have a base that contains nitrogen in it. So they often call it a nitrogenous base. Now, both RNA and DNA use a pentose sugar. What makes DNA and RNA significantly different is the actual sugar they use. DNA, which stands for deoxyribonucleic acid, uses, surprisingly enough, deoxyribose. What does the deoxy mean? That means that it is lost in oxygen that ribose sugar has. And that happens to be from carbon number 2. So you can see here on carbon number 3, there's an OH group. Over there, there's only a hydrogen. All right, so what happens is that you'll have, like I said, the phosphates and the sugars, but that nitrogenous base, it comes in 4 different versions. There is a nitrogenous base called thymine, which is typically abbreviated T. There's a nitrogenous base called cytosine, which is, you guessed it, C. Adenine is abbreviated A, and then guanine is abbreviated G. So you can see here the 4 different nucleotides that can be arranged in long strands to form DNA molecules. Now, how do I connect this nucleotide to that nucleotide there? That was part of the work that James Watson and Francis Crick tried to figure out when they were putting together their model of DNA. What they figured out, amongst other things, is that the phosphate from one nucleotide will join to the sugar of the next nucleotide. Now, the 5 and the 3 here are just referring again to the carbons of the 5-carbon sugar. So you can see you'll have a phosphate here, sugar there. Phosphate, sugar, phosphate, sugar. That's why when you form these long strands, they often talk about the sugar-phosphate backbone. Okay? So, sticking out from the sugar-phosphate backbone, you'll have the nitrogenous bases. And it's the sequence of the A's, T's, C's, and G's that determines the genetic code, just like the sequence of letters in a book determines the meaning of that book. Now, you may have heard about something called a double helix. That's because DNA doesn't form a single strand. A molecule of DNA is actually made out of 2 strands. What holds them together are hydrogen bonds between the nitrogenous bases. And one of the things that you'll have to learn in class is something called the base pairing rules, which are named after Erwin Chargaff. So they're called Chargaff's rules. And that's adenine will always join to thymine by 2 hydrogen bonds. Cytosine will always join to guanine by 3 hydrogen bonds. So what happens is that these 2 strands are held to each other. Now, something else to point out is notice up here, this phosphate is up. Over there, the phosphate is pointing down. So they are arranged in opposite orientations, like this. That terminology is called anti-parallel. This would be parallel. This is still parallel like in geometry, but because there's an orientation, they're called anti-parallel. I sometimes think of these strands kind of like a conga line. This is the person in the front sticking out their 5 fingers, their phosphates, and up here they're shaking their sugars. So you have the head of the line, the back of the line. And here I have one conga line that's heading this way, that conga line heads the other way. Finally, what do these 2 strands do? That's where we get into that double helix thing. That's where the 2 strands will spiral around each other in a double corkscrew shape. That's the double helix.